as always, we do want to acknowledge that we are joining and with UW-Tacoma in particular from the ancestral land of the Coast Salish people, and in particular the Puyallup tribe of Indians. And I think anytime we're thinking about the work that we do, thinking about the specific connections, so for shellfish and the cultural importance of those come to mind given today's topic. And just thinking about the intentionality of who we partner with and the voices we center, as always, in work that we do. So just a moment to pause and think about how that applies in your own work as well. So this is just kind of a quick overview of what to expect today. We're going to talk a lot about microbial source tracking best practices with Amy Zimmer-Faust. We're then going to hear some really exciting evolution in technology and some new insights around differentiating between different human sources from John. We'll also then hear about a new sensor with Virridy before we kind of pause for that first point of Q&A and then take a break for a brief moment since we know that it's a long day this morning. We'll then have our lightning talks, and these are really focused on some of the more regional efforts in Puget Sound, wanting to give folks a sense of what's already happening in the area before we give you opportunity to talk to all of the experts we have on today and kind of pick their brains on insights and lessons learned. But with that, I'm going to turn it over to Amy for all of the more interesting and fun insights.
All right. Great, thank you for that introduction. All right, so I am really excited to be here today and be part of this discussion. And I am Amy Zimmer-Faust. I'm currently Associate Director of the Oceans Program with the Nature Conservancy's California chapter. But I was previously a scientist with EPA's Office of Research Development and a scientist in the microbiology department down at SCWRP, where John Griffith is from today as well. So I'll be talking a lot today about kind of the things that I've learned in both of those positions and over the years of doing microbial source tracking and in different locations. All right, so I'm going to start. There you go. Let's make slides okay. So just quick overview on what I'm going to be covering today. I'm going to start with a quick overview on the concept of microbial source tracking, and then I'll be talking a little bit about some of the best practices when designing an MST study. I'll also talk about some of the common challenges that I've heard of and that I've seen come up when trying to analyze data from MST efforts. And then I'll close out with two quick case studies that highlight some of these points, as well as provide examples of novel applications of MST that I think will be relevant to the Puget Sound region. So a bit of background. I think this audience knows all too well that fecal pollution is a widespread challenge and that there are real consequences, including risks to recreators and shellfish consumers and local economies due to beach and shellfish growing area closures. And we've all made a lot of progress in terms of water quality management in past decades, but we're dealing with new pressures like aging infrastructure and more intense rainfall that continue to make pollution management really challenging. And when elevated bacteria levels do occur, pollution sources can be really difficult to mitigate because most waterways don't just have a single kind of easy source of contamination to identify. Instead, there's often multiple overlapping sources happening at the same time that make it really challenging to know where to focus management efforts. And understanding the sources of fecal pollution, which is really the key concept behind microbial source tracking, is important for two main reasons. First being that public health risk can vary by source type, with higher risk associated when the source is human, like from faulty septics or sewer overflows. And mitigation is often going to also be more effective when you really have that concrete understanding of pollution type and exact pollution source location. So having this information on source or sources allows managers to better prioritize where to apply limited resources and capacity, and then more effectively follow up and mitigate those sources. So traditionally, when we test for bacterial contamination, we use culture-based methods. And that means that we take a water sample, usually incubate it for a day or two, and then we measure levels of fecal indicator bacteria like E. coli or Enterococci. And these numbers will then tell us how much fecal pollution is present, but not where it came from. That's where microbial source tracking, or MST, comes in. MST tells us about the different sources of fecal contamination by detecting unique segments of DNA from microorganisms that live in the guts of specific hosts. So MST broadly can distinguish between kind of four main categories of fecal pollution. Human markers can tell us the sources coming from things like sewage or septics. And John in the next talk is going to talk more about methods that are beginning to advance that can even distinguish between different human sources. We also have markers for livestock, such as those from cows, pigs, poultry, that can point to agricultural runoff. There's wildlife markers that can help detect contribution from things like birds and deer. And there's pet markers that can help identify things like where dog waste may be an issue. And this list of markers continues to grow as this field advances and evolves. And John will also talk about some of the more widely used and accepted markers and measurement technologies for measuring these things. So in terms of applying these methods, I'm not going to cover every possible application. There's a lot, but there's a few examples that I'll talk quickly about. One of the most common use is for prioritizing locations for remediation. And this includes using MST to identify whether contamination is coming from human sewage, livestock, or wildlife, and then directing resources accordingly. Managers may choose to prioritize sites impacted by human sources first. That's pretty common use. But MST has also been used to evaluate best management practices. So this includes applications like where you've implemented improved agricultural practices and you want to see if you can measure reductions in cow-specific markers, really to show that those interventions are working and how well they're working. And other applications include monitoring for sewer overflows, understanding sources of contamination that drive beach closures, and investigating local hotspots of pollution. And MST is really flexible. It's successfully been applied across a wide range of sample types, including freshwater, shellfish, sediments, stormwater, estuaries, seawater, and even groundwater. And this review that I've linked here does a great job of kind of outlining studies and giving really good examples of all of these application types. So it's worth digging into if you have questions here. So how does MST work? Well, MST relies on the measurement of DNA and or RNA. So it's a shift from those culture-based to molecular methods in terms of the way samples are processed and what you're actually measuring. So first you're collecting and filtering a sample, and then that filter is generally stored or preserved before being processed, unlike with culture-based methods. Next, you have extraction, where you're isolating the DNA or RNA from the filter. And then finally, we get to measurement. And here you're testing the sample for specific MST markers. And there's multiple molecular methods that you can use, including different types of PCR and high throughput sequencing based methods that, again, I'm punting to John, who's going to talk about the pros and cons of those different measurement technologies. And one big advantage of molecular methods worth mentioning is that these provide a lot of flexibility. Once you collect and filter a water sample, that filter can actually be preserved and analyzed later. And not just for MST markers we're talking about today, but also for additional sources, for specific pathogens that can incur risk, and things like antibiotic resistance genes. So unlike traditional culture methods, which are really limited by short hold times, molecular approaches will allow you to bank those filters, and then you can actually go back and later to ask new questions as they arise. So where do these methods stand today in terms of national use and acceptance? First, it's important to note that microbial source tracking has moved well beyond the research phase. There's now really a strong foundation thanks to years of kind of research and application by academics and federal and state researchers. And in terms of EPA kind of applications, the EPA has approved 2 methods for commonly used human markers, HF183 and HUMM2, shown and linked here. And these are both based on qPCR for measurement. And these SOPs also include a lot of good information in terms of quality control, data analysis, and other laboratory considerations that are worth looking at, especially if you're doing measurement of MST methods in-house. EPA also has developed certified reference materials from NIST, which includes targets for 13 different MST qPCR assays. So this material you can get from NIST, and it gives labs the ability to calibrate your work against recognized standards, helping ensure results are consistent and reliable. And then EPA has also licensed and has patents for several other qPCR-based assays that can identify contamination from cows, dogs, and humans. And these assays have been tested for specificity and sensitivity to their host target and validated in different geographies. And then at the state level, there's also been a lot of leadership in applying and standardizing MST methods. In California, there's a state MST manual that was published a few years ago, but it's still a very relevant resource in terms of providing guidance on study design and method selection. California has also taken a big step forward, actually approved digital droplet PCR for rapid beach monitoring in San Diego, helping protect beachgoers in near real time. And then in Florida, MST has been applied to track down sources of contamination and guide mitigation actions. They also developed a comprehensive guide that may be useful that's linked here as well. And then Michigan's worth mentioning because they're now celebrating over a decade of using qPCR for routine beach water quality monitoring. So they have a really long running program and they have a lot of lessons learned about how to integrate molecular methods into day-to-day management. And these are just a few examples of things happening at the state level. And I know today you're going to hear a lot more about the great work happening in Washington as well from other panelists. So what happens when you actually go to design an MST study? Well, first, it's really important to have a robust and thoughtful study design for a couple of reasons. You know, these resources can be resource intensive, not only in terms of the cost of processing samples, but also the staff time needed for field work and lab work. So planning upfront pays off. So the first step is to develop an initial hypothesis and really think about what question you're trying to answer. If you're trying to target a specific site where contamination has been a problem versus trying to understand sources across an entire watershed, the study design is going to look a lot different. And the more specific you can be about your goals up front, the more likely your MST results are going to actually be actionable. So once you've developed a clear hypothesis, the next step is to gather and evaluate all the existing evidence that can help shape your study. And there's a wide range of data that can be useful here. This includes information that will help you think about the fate and transport of the pollution sources that you're targeting. So this includes environmental and physical conditions. For instance, if you think you have transport through sediment, understanding something about the sediment type can be really useful. In sandy or coarse grained sediments, you'd expect faster transport versus organic rich sediments. And that's just one example. This also includes pulling down climatic and hydrologic data that's going to be really useful for telling you something about how quickly you can expect impact, how that pollution source moves through your system, and where you're most likely to be able to detect it. You also want to think about data types that tell you something about potential sources. This includes getting as much data around land use and infrastructure as possible, and then mapping out potential sources as best as you can. This includes point and non-point sources and other relevant features too. And then looking at historical water quality data, and not just FIB, but any water quality data that you can get your hands on can be really useful to examine spatial and temporal trends and provide important clues on when and where you expect problems to occur. And then if there's any sound or sensor data, if there's continuous monitoring, even for things like salinity or temperature, that can be useful. And then Alex from Virridy is going to talk later about E. coli sensors too. And all of these continuous monitoring efforts can be really helpful to highlight spikes or patterns, which could then trigger more targeted MST investigations. So really complementary. And then lastly, just visual reconnaissance and surveys can be really helpful. Going out and actually looking at your sites and talking to folks, that local knowledge about conditions that may impact pollution sources and transport can really be your friend. So the goal at this stage in your study design process is to assemble the most complete picture possible to help make sure your MST study is as targeted as it can be. And then the next step is just using all of that information that you've gathered to refine that hypothesis and really drill down on what your study objective is. All right, and so last I want to talk a bit about designing the study itself. And this is where you're thinking about the when, where, and what to sample. So when to collect samples, timing is absolutely crucial. You want to consider whether high fecal indicator bacterial levels have in the past corresponded to things like particular flow events or rainstorms, whether they occur year-round or have some seasonality to them, and look at tidal cycles as well. That can be an important factor too. And then the where to collect samples. You'll want to sample near known or suspected sources. You want to keep in mind dilution and mixing. So the further downstream you go or the further away from that pollution source origin point, the harder it may be to distinguish individual sources. So really think strategically about where you can get the clearest signal. And it can also be useful here to include reference sites. So places you aren't expecting contamination for verification. And then what to collect. Finally, you want to decide what and how much to collect. You want to make sure you gather enough samples for statistical significance, and you also consider redundancy. For example, if cows are the primary concern for you as your kind of top pollution source, you might want to consider both a general ruminant marker and a cow-specific marker for confirmation. And since field efforts can be expensive and difficult to replicate, it's often worth collecting and storing replicate filters for potential future analysis so you can go back in and measure other things. So once you've designed your study and started collecting samples, the next challenge is what to do with the data and actually interpreting MST results. You know that this isn't always straightforward. And that's for a couple of reasons, just a couple that I'm listing here, but there's more to it than this. So first, unlike traditional fecal indicators such as E. coli or enterococci, we don't yet have established risk-based thresholds for MST markers. So that means the results can't be directly translated into health risk in the same way. So how much is too much is a question we often ask ourselves. Second, MST studies often show a high frequency of low or non-detections. And this doesn't always mean that the source isn't present. It may simply be below detection or diluted or transient. So the timing may not have lined up for you to detect that source. And then third, environmental persistence and decay can complicate interpretation. Some markers break down quickly while others may persist in the environment for longer. So given all this, what can you do? Well, first step, I'll talk about the challenge that there aren't any established thresholds that tell us when a marker level is too high or poses a health risk. Despite this, there's a lot of very productive ways to still use MST data. We can use the quantitative information to make important comparisons. For example, we can look for hotspots, areas where marker concentrations are clearly higher than surrounding sites, or where we see a gradient that points us towards a potential source. We can also overlay MST results with traditional fecal indicator bacterial levels. So we might think about at sites where bacterial levels are the highest or days where we had exceedances. MST can help tell us and help us pinpoint sources that are contributing on those days and the most often. And finally, there's exciting progress in the research space to address this challenge too. Over the past few years, there's been several studies that have applied quantitative microbial risk assessment to actually develop risk-based thresholds for certain markers, including the commonly used HF183 human marker. So while there aren't formal standards yet, these studies can provide valuable benchmarks that can help us interpret results in a health relevant context. So I've included a few of those studies here, and you can see that the estimated risk-based thresholds really vary in these studies from about 100 to 500 copies per 100 mil, and those levels are dependent on the source and where the exposure occurs. So the second challenge, lower non-detectable results can be really common in these studies, so it's important to plan for them. First, just trying to reduce false negatives as much as you can be key here. And to do that, you want to sample as close to the source as possible and collect enough samples under the right conditions when you think that that source will be present. And then when looking at the results, don't just focus on concentration. It can also be helpful to visualize how often markers are detected. The figure on the upper right of this slide does a nice job of doing that. This is a heat map that's showing different MST markers, and you can clearly see both where they were detected, and then the color indicates concentration. So this can be a useful way to visualize data when you have a lot of non-detects or low-level detections. It's also really important to clearly define and then communicate detection limits. If you're using a contract lab, labs may report these differently. So just making sure that you get this information so that you can compare between studies, I think is critical. And then finally, it can be helpful to pick data visualization methods that allow you to see the range of concentrations and distribution at different sampling sites. So methods even like box plots are shown here on the bottom right. These are violin plots that show log scale data, can be really useful because it allows you to show kind of all of the data and the distribution of the data that you're getting at individual sites. And that can help kind of normalize some of the variability that's intrinsic to these measurement methods. And then lastly, there's concern about how long these markers last in the environment, how differences in environmental decay and persistence can complicate measurement. And degradation or decay of these markers in the environment can result from lots of things, but inclusive of some things like sunlight and predation and even sedimentation or settling out of the water column. So sampling as close to the source as possible, again, also targeting those conditions associated with transport of the source can limit opportunities for environmental decay. So it can kind of be out there sampling when the pollution source is fresh. That can be a helpful tool. And then secondly, just thinking about the appropriate markers and including some redundancy by selecting more than one marker for the pollution source that you're most interested in can be helpful. And there's lots of studies that exist that provide more information on decay rates of different markers, so you can choose accordingly and kind of better understand what your marker is likely to do in the environment that you're testing. And linked on this slide is a really nice review of a lot of these concepts that this slide has addressed. All right, so now that we've gone through some of those kind of tips and tricks when both designing your MST study and analyzing the data, I want to walk through two quick case studies where these approaches were applied. And I'm going to start in San Diego, where they experienced frequent beach closures from really complicated sources. So this first project, this was an MST study done at the US-Mexico border. And there's been a lot of headlines about these issues occurring here that you may or may not have seen. And this is, an ongoing and complicated problem. And it's an area where there's multiple point and non-point sources of wastewater pollution. So the primary of this study was really to understand and kind of isolate the impacts from one specific wastewater treatment plant, the San Antonio de los Buenos plant on coastal water quality. And this plant is located about 6 miles south of the US-Mexico border. And at the time of this study, this was a constant source. It was only treating to partial primary, so just settling ponds that were mainly defunct. So this was pretty close to raw sewage that was being dumped into a stream that terminates at an ocean outfall. So it's kind of known and predicted that this effluent could sometimes be making it north to South County, San Diego beaches, but there wasn't a lot of good data at the time to support this. So the study's main objective was to quantify the extent and impact of pollution from that faulty plant and see really how far up coast it could get. And so the geographic scope of the study, we focused on the binational coastal zone near the US-Mexico border, and we wanted to bracket that treatment plant. So to get the project started, the first thing that we did was to convene and meet with stakeholders and potential partners. We actually had an in-person project kickoff in Imperial Beach, so local, and that was really key to kind of understanding what other efforts in the region were ongoing and to getting partners on board. And this meeting followed a lot of individual conversations that we had prior to garner support and really encourage attendance. And the meeting included academic partners, both from US and Mexico, included local municipalities and utilities, and included federal agencies like the EPA and the International Boundary and Water Commission. And so from this meeting, we were able to both garner support for the work, build awareness, and build really critical partnerships that helped in the success of these efforts. So moving on to the planning phase. So after this meeting, we went to design the study, and we wanted to make sure it answered our study question, but it also produced useful data to other efforts that were ongoing in the region. And that included modeling that was being done at the time at Scripps to look at the same pollution sources. So again, we kind of talked, we looked at all the data that was out there. We had discussions with the modeling team, but we also looked at the historical FIB data, and we looked at ocean conditions associated with beach closures in San Diego. And the modeling data was helpful here to get a feel for those ocean conditions associated with northern transport from that wastewater treatment plume. And what we found was that swell direction and wave magnitude and actually wind direction mattered a whole lot. When wind was blowing offshore, the plume was likely to move quickly out to sea and not be transported up coast, so we wouldn't be able to measure it in samples that we collected in the beach. So we thought about all this when we were designing our study. And so we worked with those partners that we had been meeting with to collaboratively come up with a study design to decide when and where to sample. And we decided to sample from just north of that problematic wastewater treatment plant, 14 miles up the coastline. And we targeted south swell sampling events that were related to northern transport of the plume. And we also paid attention to wind direction as well. And we sampled multiple days per event to increase our chances of capturing the sewage signal and to better understand timing of the plume. Worth mentioning is that our sampling design changed a lot from where we started and kind of what we had anticipated. This was a bigger geographic scope, and we hadn't planned on sampling on consecutive days per event. And this was because we had learned a lot more about the conditions associated with detection of that pollution source that we were interested in as we dug into the existing data. So what did we measure? We measured in enterococcus, and then we also measured 2 human markers. Again, human was the primary source that we were interested in, so we wanted some redundancy. We measured HF183, one of the more common human markers, and then LACNO3, kind of a newer human marker. And we had this ideal sampling design and all of this lofty field work that we wanted to do, but next we had to contend with our budget. And so what we did here is we got a bit creative and we really worked with partners to be able to do the study that we wanted. First, we worked with Tijuana Waterkeeper. They're a really fantastic citizen science organization operating in Mexico, and they helped us sample south of the border. And then north of the border, we also worked with the Imperial Beach lifeguards. They were really enthusiastic and they wanted to better understand where those sewage smells that they'd been smelling were coming from. And they drove us between sites, which condensed our sampling time. So this allowed us to collect samples on the same day from this 14-mile stretch and then process within holding time constraints, kind of filter the samples within holding time constraints. So what did we find? Well, we were able to actually quantify the HF183 human marker about 14 miles north of that problematic treatment plant. And the dilution that we saw closely matched and helped validate what the Scripps model had predicted. So the bar plot on the right, this is showing the HF183 human marker concentration from just south of the plant, at the plant, and then up coast. And we were able to kind of get this nice gradient because we thought a lot about and targeted conditions that allowed us to really isolate impact from that treatment plant. And we picked days where it was most likely that the effluent was flowing up coast and would hit the samples, the sites that we were sampling at. And just a note, similar to the Puget Sound region where there's a lot of fantastic modeling work in progress, MST methods can be really complementary in helping validate those modeling efforts, and those modeling efforts can be really useful to help understand where and when to sample, like in this study. For example, if you're concerned about the impact of septics on shellfish growing waters, knowing something about circulation and mixing within your estuary can be useful to targeting when you want to go out and collect water samples for MST analysis and when most likely those sources will occur and show up. So lessons learned that apply to this audience. First, just engage early. I think, the fact that we had talked to stakeholders and collaborators from the start was really useful here in both helping us design a robust study and helping folks agree and kind of believe in the results that came out. We also leveraged local capacity. We were aware of other monitoring efforts that were going on in the region, and we really utilized those to be able to kind of keep the costs down on the field sampling that we wanted to do. And then I already mentioned this, but really this MST data helped build trust in other modeling efforts and kind of understanding of the impacts of this wastewater treatment plant. It was nice to have a robust data set that people could turn to. Then last, just the ocean conditions were key to explaining MST results. I showed the good days that we picked a sample where we got everything right, but there were a few days where we sampled and we didn't see this great, didn't detect the HF183 markers because we didn't have the ocean conditions. They weren't lined up. So our understanding of what is impacting transport of the plume really helped us be able to contextualize when we were able to measure HF183 and when we weren't. All right, my last case study, and I'm going to try to go through this quickly, was one that was completed in the Tillamook Bay watershed, evaluating sources of fecal pollution and nitrogen in parallel, using MST as one of the tools. And this was just recently published in Water Research, which I've linked here if folks are interested. So Tillamook Bay is really a productive rural watershed on the Oregon coast, and it supports dairy, timber, and shellfish aquaculture. But unfortunately, it also has a history of water quality impairments, including for temperature, dissolved oxygen, fecal bacteria, and elevated nutrient levels. And the potential pollution sources here include agriculture, there's extensive dairy operations in the lowlands, wildlife, and human, both for municipal and on-site wastewater treatment systems. So it's a complicated watershed that makes both nutrient and fecal pollution management challenging and complicated. So for this study, the main objective was to quantify sources to Tillamook Bay. And at this time, there had been some efforts to improve water quality in the watershed that hadn't translated to improvements in the bay. So folks really wanted to better understand why that was. So again, before we started sampling, we wanted to make sure we had a good understanding of the watershed and how different land uses might affect water quality. So we pulled together maps and data layers that showed things like where point sources are located, where animal operations are, and how land cover is changing from the forested areas that are in the uplands of this watershed to agricultural to more developed land. And then we also look back at years of historical water quality data to really understand what environmental factors might be linked to elevated fecal bacteria levels. So when we went to pick our sampling sites, we were able to place them in locations that would kind of bracket or capture the influence of both the point sources and the non-point sources that we were interested in. We also chose our sampling dates to cover different seasons so that we could test weather patterns shifted through the year. In total, we monitored 15 stations spread across the 5 major tributaries, moving from the forested headwaters down into the agricultural or more developed areas. And finally, since these rivers are influenced by tides, we made sure to take that into account in our analysis, trying to target low tides, but also paying attention to salinity in our samples so that we could take into account tidal shifts and make sure that that didn't throw off or influence our results. There you go. All right, so in terms of what to sample, here we chose MST markers that targeted sources of interest. And then for the most important sources, cow and human, we had more than one marker that we measured. For cow, we measured CALM3 and CALM2 and the general ruminant marker as well. And then for human, we measured those 2 EPA human markers, HF183 and M2. And then on the same sample, we took the filtrate to look at stable isotopes of nitrate and nutrient levels, since for this study, we were interested in the co-occurrence of pollution sources that contributed to both elevated nutrients and fecal bacteria levels. What was neat was we were able to actually split the same sample. So this field work kind of was able to give us clues about both things. And on the filter, we conducted MST analysis. And then on the filtrate, we conducted analysis to look at elevated nitrate levels. So putting it together, and I'm not going to go through all of the results from the study, I'm just going to highlight a few things. What we saw is that as the sites moved from forested to pasture, we saw increasing levels of the cow marker at similar magnitude to increases in the nitrate stable isotope signature. And I'm just showing that for one of the rivers, one of the 5 rivers that was sampled, for the 5 sites along that river, moving from upstream to downstream. So we saw that the cow marker, which I think is in blue, sorry, that's not better labeled, and then the stable isotope for nitrate, which is, I believe, pink, increased at similar magnitude as we moved downstream. So this really helped point to agricultural inputs as sources of both nitrate and fecal pollution in this watershed. And this was really useful. In this watershed, there's a lot of nitrogen fixing red alder in the uplands that can kind of complicate where that elevated nitrogen levels are coming from. So it was helpful to have these multiple lines of evidence. And they also correlated when we looked at land use patterns too, which was really nice. And I'm going to skip this one, but it's in here in the deck if you're interested. This is, again, how we kind of put all of these different lines of evidence together to identify at what sites human sources were most important and at what sites agricultural sources were most important to guide downstream management and mitigation. All right, so in terms of lessons learned from this study, I think we leaned really heavily on spatial analysis, which was really useful. We used GIS and we did a lot of analysis up front to draw upstream catchments for each site. And then we were able to summarize the land uses for each site and test those land use patterns and how they lined up with MST results. So that site level source kind of profile allowed us to better pinpoint likely drivers of contamination. I think it's also really important, especially for estuarine watersheds, to remember that these systems are highly dynamic. So in tidal rivers or other complex environments, thinking about the timing of sampling and the conditions make a big difference. And we designed our strategies to account for that. Third, data can be a really powerful bridge. This was a lesson learned both the cross-border study and this one. Here we heard from the Oregon DEQ that just having these MST data was helpful in being able to work with farmers in terms of pollution abatement because it showed that they were relying on the data. They didn't have preconceived notions about where they thought these elevated bacteria levels were coming from, that they were looking holistically at multiple sources. And then lastly, just considering catchment-wide dynamics and complicated watersheds like these. So, like I mentioned, prior to this study, there had been challenges in relating improved water quality downstream to things that were happening in the watershed. So here we were able to look for hotspots of pollution and then also think about how those hotspots were being translated downstream and where were the locations that were most likely to impact Tillamook Bay water quality. And so I think that helped, again, direct management efforts. So the last thing that I'll leave you with before handing it off to John is just that, MST really works best when you're confirming what you already expect from monitoring or modeling, and then you're using it to challenge assumptions made when the results tell you a different story. So not applying these methods in a silo, but instead plugging them in as a tool in relation to all the other work that's happening in your system, I think can really help interpretation and contextualization of the results that you get. So thanks so much for having me, and I'm happy to answer questions or follow up beyond today's workshop as well. It's always fun to talk about this stuff.
Awesome. Thank you so much, Amy. Great insights. And we're going to have John give his presentation and then Virridy, and then we'll pause for questions. We want you all to have kind of a comprehensive picture across these talks. And I should mention while John is pulling that up that Amy very kindly is doing this presentation during her maternity leave for you all. That's how much she loves microbial source tracking.
All right, well, so I'm John Griffith. I head the microbiology department at Southern California Coastal Water Research Project. I'm also the coordinator for molecular technology here. I've been doing source tracking work for well over 20 years. And so I'm just gonna kind of walk you through the background of where source tracking came from, kind of get you up to speed on the state of where we are in terms of the technology, and then talk about the latest kind of technology that we're working on. Okay, so with the origins of microbial source tracking, where did it come from? Well, so folks have had this problem with fecal indicator being non-specific for lots of years. And so initial ideas on how to tackle this were based on the idea that microbes and their hosts evolved together. And so there should be traits or genetic features in the microbes that we can use to match isolates to their host. And so that meant you had to build a library of source isolates. And you can imagine that this gets tedious, and the library would have to be really big to encompass everything. But typically 1,000 isolates is a pretty big library for this kind of work. And so they focused on phenotypic methods, and these are things were also very tractable for students to do. So you can see the appeal for professors using things like antibiotic resistance analysis, with the idea that humans take a certain suite of antibiotics. There's a certain suite of antibiotics used in livestock production, and so on. Then other things like carbon utilization, using things like BioLog, which is a commercially available multi-well system that makes it easy to do that kind of work. And then genetic methods from the food safety sector, which are still in use, especially PFGE, although it's being supplanted by sequencing now. Also ribotyping and box PCR, probably some things that none of you have ever heard of, right? And so by 2003, there had been multiple methods that had been published, and of course the papers said they all worked. But the way they worked was, if you took an isolate out of a library and you challenged it, could you reliably find it back in the library? So they use things like discriminant analysis. And some folks were using this actually commercially. There was actually a guy at UW that was doing PFGE that had mainly worked in foodborne pathogens, that was using this to profiteer, because managers were desperate, because they were under pressure from regulators, to find out what the source of contamination was. None had been independently tested. And so when I first came to the agency here, we did a method evaluation study with money we got from the state, and we showed that most of these library-based methods were no better than coin flip at identifying the sources. They just didn't work. Okay, and so for about the next 10 years, things continued to develop. Host-specific genetic markers had shown promise in that initial study, and this was based on work that was done by Kate Field at Oregon State and her graduate student Ann Bernard, and they developed source markers for human and dogs and cattle, and but they were all just run them out on a gel and see if you got the right size fragment. And so they were just presence absence. And so over time, the library methods completely disappeared. I quit seeing any students with posters at the ASM meetings with them. And researchers turned to developing these new methods, making them quantitative using qPCR. And so by 2013, there were greater than 50 source-specific methods for different fecal sources that have been published, and many competing ones, especially for humans and cattle, which seem to be the biggest sources of concern. But managers didn't know which to pick, and so kind of it depended on which university that you decided to contract with, because that's who was doing this work, as to which markers you ended up using. And so once again, an independent method evaluation study was needed. And that we actually did a method evaluation study. It was part of a larger project to be able to bring source ID methods to managers so that they could do them in their own labs. I'll talk about that briefly in a few minutes. So the first thing you need to do for the method evaluation, challenge the methods. So we sent out 64 blind samples, combinations of 12 different sources. We had 41 methods, 27 participating labs, and most methods were run by multiple labs. So a lot of the labs ran not only their method, but one of the other methods from one of the other labs, because we needed to know if the method was transferable. So here are the sources. And we had 3 human sources. That was the thing that was most important to folks. So individuals, sewage and septage, a number of livestock, dogs, and then geese, deer, pigeons, and chickens. And so the way we did this is we collected samples from all over Northern and Southern California. We had 4 participating main labs, Allie Bain at Stanford, Trish Holden's lab at UCSB, Jenny Jay's lab at UCLA, and our lab here at Squirt. And you can see the different markers correspond to what they collected. So graduate students went out over a couple of days, collected all these, and drove them down to Squirt, where we were going to make the samples in just south of Los Angeles. And so here's a couple of the stalwarts checking in samples, and you see these tubs down here. These are the let me use my cursor here. These are the individual human samples. So most universities have an internal review board, and it turns out that human poop is considered tissue. And so they could not collect samples for human. So we had the graduate students carry their samples internally. When they showed up here at our lab, we handed them a Tupperware tub, and so they all participated in the study in more than one way. Okay, so we took these and basically combined them with artificial water. We stirred them to mix the liquids or homogenize them in a blender, screened them to get out the chunkies in the corn, measured enterococcus by qPCR to kind of standardize the dilution, and then put equal volumes at each sample in together to create composite solutions, or use them as is mixed and or diluted. There are greater than 6,000 individual filters or water samples created. It was 7 days of near continuous work. I had students that were shuttling back and forth from the airport from Stanford. We had carpools from UCLA. I had professors coming down and helping. And then we distributed them across 7 states and 6 European countries. And so here you can see the samples being weighed out on the top and the blender crew on the bottom. You notice they're outside. There's a reason for that. No one wanted that in the building. And here's what folks looked like when we started. Everyone was happy and filtering away. And here's the aftermath. And then at one point we had to enlist child labor. Not really. So this is Jenny J from UCLA. She actually brought her kids with her because she wanted to come help and she didn't have a babysitter. So they observed. So here are the participants. I'm not going to go through them all. I'm just showing you this to see that so you can see this is pretty much everyone that's still doing source tracking. If you look at the papers in source tracking, you'll see that these are still the major players, with few exceptions. Classes of methods, we had presence-absent methods, we had quantitative methods for single sources, and we had some community methods such as phylochip, that may also provide information about relative concentration in the sample, but they could do multiple sources at once. So our criteria, could they identify the source? Could they identify the dominant source? And then how repeatable were they? And it turns out what we ended up in the end is probably about 20 binary acids that met our criteria for specificity and sensitivity, and about 10 quantitative assays, some of which you heard Amy talk about. And then in addition, all the community methods were excellent for deer, which we didn't have a marker for, and phylochip could pick out chicken, which was quite interesting. All the technical aspects of these are at the end. There's a citation for the paper that Allie Bain is the author on for this. But the outcome was we identified methods that were both specific and sensitive for 5 major classes: human, dog, pig, cow, and gull. And what got something that is unusual in the scientific community, consensus across all the key scientists And these folks all have big egos. So they all agreed which markers did best. So this was great. We had a dedicated issue from Water Research, I think 17 different articles. And so we were able to use that to put into our source identification manual, which was one of the goals of this effort. And so we not only evaluated the methods, we test drove them, and we did demonstration projects at 3 beaches. Just an aside, one of these, Amy was talking about the multiple sources and teasing them out. We had one beach in Santa Barbara that was chronically contaminated, and they had found leaky sewers and fixed them, and they thought they had it all cleaned up, and they still had a bacterial problem. And so when they used markers, they found a dog signal, but they couldn't find any dogs on the beach, and this beach had a creek that ran into it. And so one of the graduate students decided to do some reconnaissance, which is something we suggest in the manual, and went looking upstream for dogs and didn't find any, but he found dog poop. And he's like, well, where's this coming from? And as he's walking along, all of a sudden dog poop comes flying over the back wall of one of the residences. And so it turned out that some folks were throwing their dog waste over the fence, and it was getting into the creek. And they were able to remediate the problem by doing a public service flyer and putting it on all the houses that bordered the creek. And it actually helped to abate the problem. Okay. So the microbial source tracking manual basically goes through the steps that Amy talked about. It went through a whole lot of review, including by EPA. It's a tiered approach, hypothesis driven, which is really important. You need to know what your questions are going in. As Amy said, start with the less costly methods, the indicators, and then as you refine the problem, then use the more expensive methods. It's really easy to bust your budget with molecular markers early on. Okay, so what about molecular methods? Well, there are multiple gene amplification methods that can be used for microbial source tracking. They all kind of work the same way. They amplify up a particular gene that's diagnostic of a particular source and a particular kind of bacteria, and to a level that you're able to quantify it or at least see it. qPCR is by far the most common. So all those quantitative methods that I showed you from the evaluation study are all qPCR methods. EPAs are all qPCR methods. But qPCR is old technology at this point. Digital PCR uses the same primer probe sets, but it's more sensitive and it's less prone to inhibition. All of the qPCR methods can generally use the same primer and probes that you use in any assay, not just for MST, as long as you optimize them. There's no need for reference material, which really makes it more robust. So you don't need a plasmid and you don't need a standard curve. We use the Biorad droplet digital PCR. That's the most common variant because it came out first. But QIAGEN and Thermo also have chip-based digital PCR. And digital PCR is actually more easily multiplexed than qPCR, which means you can test for more than one source at the same time in the same sample, and that actually can save you money. It's not very easy to do more than 3. There are some machines that tout that they can do 6, but you lose sensitivity as you multiplex. So there is a limit to how much you can do, but still a big step forward in the quantitative part of PCR from qPCR. Okay, there are also other molecular methods. There are isothermal methods such as LAMP, loop-mediated isothermal amplification, NASBA, transcription mediated amplification. The last 2 are RNA-based methods. all of these are have some advantage in that because they're isothermal, you can actually do them pretty easily in the field. You still need to run a standard curve. And so they aren't so widely used. They have not been included in an evaluation study. And then the last thing I wanted to just touch on, it's really only been used, it's been talked about for source tracking, but folks have not gone in on it yet because it's still pretty expensive, but it's CRISPR. This also can be used for gene editing. It's really sensitive and quantitative when digitized and coupled with a pre-amplification step. It also is an RNA method. There are multiple medical diagnostic tests on the market. It's just beginning to be explored in the food safety sector, but high potential for environmental MST. We don't know how it will do in the kind of dirty samples we have. So that's going to have to be tested. But crassphage, which EPA has been pushing as a viral source tracking marker, may be an opportune target. These phages primarily infect Bacteroidetes, the same kinds of bacteria that are used for source tracking. And CRISPR actually targets the insertion points of these phages in the bacteria. And so that's what gives them their specificity. And so you can use these palindromes, these repeats of base pairs in the genome as your target. And so I'm really interested in this. And I'm looking for it to get cheap enough for someone to collaborate with me so that we can start trying to use it for MST. Okay, last thing we want to talk about is differentiating between different human sources. So single source human markers don't differentiate. HF183 can't tell you where a human source came from, whether it's an individual human, whether it's septage, whether it's from a sanitary sewer. And so this is the beginning of trying to figure out how to differentiate between human sources. So in this case, we actually used biofilm from the inside of sewer pipes. It turns out that the inside of a sewer pipe is a pretty specific microbial environment. And this was based on work that was first done by Sandra McClellan at University of Wisconsin-Milwaukee. And so we wanted to see if we could use it in Southern California. And so one of the things we see is in wet weather, we see a huge load of HF183 every time it rains, and we don't see it in dry weather. But we can't distinguish in the stormwater between the different human sources. And so the goal here was to develop and test the tool in the San Diego River watershed. What you're seeing over here is basically a volumetric plot of HF183 in the watershed. Oops, back here. So this is the main stem of the river. These are all tributaries on the side, and this is the load of HF183. We basically put out flow meters and also took composite samples so that we could measure the load at each of these places. And so this is for one storm during that time. So the first thing we had to find out is, are the biofilm microbial communities different between sanitary, sewer, and storm drains? Second is, how sensitive and persistent is the biofilm community in the stormwater? Can we see the signal? And then, do we see these signals in wet weather samples? And I'm just going to give you the answers. Yes, they are different between sewer and storm drains. They're persistent across years and seasons, sensitive enough to apply to stormwater, and yes, a widespread dilute signal is seen in stormwater when it rains in the San Diego River watershed. So here's how we went about looking at the stormwater and the sewer and getting the biofilm. All these are biofilm sites. The yellowish ones are storm drains. The green ones are sewers. We actually went out with the agencies and popped manholes. We developed a device connected to a really huge industrial size peristaltic pump that my technicians dubbed the scum sucker. And we take the scum sucker and we actually suck the biofilm off the surfaces in the sewer and the storm drains, along with a nominal amount of water. And so here you see a cluster plot of the sewers on the left and the storm drains. So this is from 16S sequencing. So this is basically sequencing all the bacteria in the sample on an Illumina sequencer, and then using source tracker and clustering the results. And what you see is that we get a nice distinction between storm drain bacterial communities and sewer bacterial communities. Okay, so we need to know, we need an empirical way to quantify this. And so what we did was spike aged stormwater with the sewage biofilm. And what that allowed us to do is figure out what proportion of HF183 we could measure. And so you can see HF183 in 100% stormwater, it's about 10 to the 7. At a 10-fold dilution, about 10 to the 6, 10 to the 5, and 1,000-fold dilution and 10,000-fold dilutions, 10 to the 4, 10 to the 3. And so what you see is that we're able to empirically measure what our signal is in the stormwater. And so this allowed us to do some experiments looking at loading. And so how sensitive and persistent? Well, we selected 13 sites along the main stem and major tributaries. We sampled 3 storms using grab samples, and then we measured HF183 in the microbial community biofilms. We used HF183 to confirm there's a human source in the stormwater. And then if we found a sewer biofilm, we determined the strength of the signal. We used the sensitivity experiment I showed before to calibrate the sewer biofilm community abundance to the experimental dilution. We categorized the sewer biofilm proportion in the experiment and stormwater samples using source tracker, which is essentially a pattern matching algorithm developed by Mike Sadowski's lab at University of Minnesota. Actually, Amy used it in that study she talked about along the US-Mexico border previously. And then we compared the proportion of stormwater samples to our experimental samples to estimate the strength. And so here's an MSDS plot of our samples and how they clustered. Here you see our experiments, 100% sewage, 20%, 10%, 1%, and so on. And so we see a nice gradation as we get from sewage biofilm over to stormwater. And then here is our storm drain samples up here. So overall, we were pretty pleased with that. And so when we looked at the strength of the biofilm, what you see here are these black bars are our test sites. And here is our experiment, or sorry, the black bars are our experiment. These are, I'm sorry, I had it right. The gray bars are experiment. So here's 1% sewage, 0.1% sewage, 0.01% sewage. And then here are our test sites, this Murphy Canyon Marina. And you can see we peter out as we go along. This actually corresponds well with what we saw in our volumetric plots. So here's Murphy Canyon. Here's Marina. But we found something that was interesting. This is Eucalyptus Hills, and we had this big HF183 signal, and we're like, oh no, maybe it doesn't work, except it turns out Eucalyptus Hills doesn't have any sanitary sewers, is completely unseptic. And so this actually turned out as further proof that we could use sewer biofilm to look at sewage in stormwater and quantify it. And so that was pretty exciting for us. So the highlights, we found sewer biofilm in nearly all the stormwater samples. The sewer biofilm was pretty dilute, so less than 0.01% to 1%. We measured HF183 in the storm samples. The concentrations range from 10 to the 1 to 10,000 per 100 milliliter. So quite a lot. Here are the citations for what I presented, because I obviously did not have enough time to present everything. And this is a link to the Microbial Source Tracking Manual if you're interested in it. This is the biofilm paper. Here's that 2003 study that basically debunks the library-based methods. And if you have any questions, or you'd like to know more about what we do, or just talk about source tracking, email me. I'm happy to chat with you.
Awesome.
And with that,
thank you so much, John. And as we move to our next presentation, again, after that, we'll have time for a Q&A. I'm seeing lots of good discussion in the chat. But while Alex and Whitney are pulling up slides, John, are you still looking for a septic system sample?
we are looking for septic system samples.
Do you want to just share a little bit of what you're looking for? We've got an audience here who does a lot in this space, and sometimes matchmaking opportunities happen. Yeah,
so it turns out that that people are actually really worried about giving up their septage. In fact, when we did an initial study in Malibu one time, we don't do press conferences, but we were working with the county and they did a press conference. And Malibu, as you may know, is home to the rich and famous. And after the county's press conference, they interviewed Pam Anderson on the radio, and she evidently was quite concerned. The county had said we'd be able to follow the signal back to the source, and she said, "They're not getting any of my poo in my septic tank." So she was quite worried, right? So people are worried, and septic systems on private property. So the only access we really have to septic systems is if we collect from pump trucks that are coming into the sanitation district. And that's not really what we want. We actually want material from the non-sludge portion of the second part of the tank where the solids sit. So basically the leachate that's going to go out the leach tube. And the pump trucks pick up all the sludge and everything else. And we think that's probably not the same community that we see in the water that's running out the top. So that's what we're looking for. We'd like to do the same kind of thing that we did with the biofilm with sewers, with sanitary sewers. So we'd be sequencing that and then looking to see if we could find that system, that signal that we characterize from different septic tanks in stormwater.
Awesome. Thank you, John. I know a lot of folks on this call would be very interested in differentiating between septic and others. And so if anyone happens to have projects that are digging up septic systems or have opportunity to get to that, please feel free to reach out. But with that, I'm going to hand things over to Alex and Whitney.
Hello, folks. I really appreciate your time, and we're certainly following along. We actually many months ago engaged with Amy on the Tijuana example that she shared earlier in this presentation. My name's Alex Johnson. I'm the Chief Strategy Officer for a company named Virridy, which is actually a spin out of CU Boulder's Global Engineering and Resiliency Center. Virridy actually works a lot in drinking water delivery in 5 African countries, and as such, we've been researching a lot of distributed monitoring solutions for fecal contamination, and are now kind of commercializing and launching one of those solutions. Obviously, not just for drinking water projects in the developing world, but we're also seeking and testing these sensors in the US, in Europe, et cetera, as you will learn. So, in terms of this workshop, we just want to bring everybody up to speed on our progress there and this sensor type and what we're seeing in kind of the beta testing with partners, just so you are all aware of another tool in the toolbox that's coming online that we're pretty excited about. Let's see. So here's the loom. It's about the size of a Chipotle burrito. It's meant to fit inside of a PVC pipe, as you can see on the right there. And basically it uses fluorescence of organic matter in water passing through the device. It flashes it with an LED and then picks up the relative fluorescence coming back. And from that, we can quantify levels of E. coli, chlorophyll A, FDOM, a couple different things that my colleague Whitney is going to touch on in more depth here shortly. But the nice part is it's a very simple device, and we connect it to a machine learning network so that we can kind of improve the data after we get it and modify for battery life and time since last cleaning, et cetera. So there's no moving parts, and that allows us to produce these at pretty low cost, which is part of why we're excited about this. We can manufacture these for around $1,000 a unit, which is dramatically cheaper than other things that we see on the market for fecal contamination, for E. coli specifically. And so we're hoping that we can fill kind of a niche here in the sector in both developed country applications and developing country applications because of kind of the low cost and the distributed nature. And this is a unit that can, connect to cellular networks, upload data. We currently have it usually on a 15-minute collection event timeline, and the battery lasts 6 to 12 months based on how much data how fast you're submitting data. So that's kind of the innards, and what that then allows us to create. If you go to the Virridy website, pretty easy to find this dashboard. There's, I think, 20 of these out in the world. And as I said, some in urban waterways like Chicago and Boston and Paris, and then some in boreholes and drinking water pipes in Kenya and Rwanda and a couple other places as well. And it's obviously giving you every 15 minutes what you see on the screen. And Whitney will dive into how sensitive we can get it and at what standards. So I'll pass over to Whitney.
Awesome. Thank you, Alex. So I'm just going to first give a little bit more of an overview of the mechanisms related to the technology. So the Loom sensor uses a fluorescing amino acid in the water known as tryptophan. And so any fluorescing organic matter in the water that fluoresces at that defined wavelength is known as tryptophan-like fluorescence. And it has been well defined as a proxy or indicator for microbial activity. So as Alex mentioned, these readings are able to be taken in real time, which is significantly different than typical microbial monitoring that requires sampling and then lab analysis and incubation. So you only get samples with any sort of data on quantification of contamination 24 hours later. So the sensor takes fluorescence, essentially fluorescence intensities, and fuses those into a machine learning analytical model that takes into account all of these other factors influential to microbial contamination, including, as Alex mentioned, time since last cleaning, which accounts for biofouling, as well as turbidity, and then in the future things like stream flow, rainfall data, land coverage data, all which are influential to the types of microbial contamination and the magnitudes. As Alex also mentioned, Virridy is a spinoff of the research done at the Mortensen Center at the University of Colorado Boulder, where I'm also a PhD student. So these technologies are developed by the company and then further validated and applied into contexts in the university, which allows us to get a lot broader of research opportunities to validate and apply the sensor. To get a little bit more into the features of the sensor itself, it quantifies actual E. coli concentrations, and by that, the model right now either establishes a threshold of above or below, like a binary prediction model, or in alignment with the WHO risk categories for microbial contamination of drinking water. So that is dependent on the context. But the key aspect is that it provides quantification in E. coli, which other fluorimeters that monitor tryptophan-like fluorescence typically provide units of relative fluorescence, which are not super meaningful when it comes to microbial contamination. Beyond this, the machine learning model utilizes and learns with the data, so it doesn't require regular calibration, and it is designed to be continuously used in situ. So it currently uses a battery, which has battery life from 6 to 9 to even 12 months, depending on sampling frequency. Likewise, it has the potential to be connected to a solar panel, so there's no maintenance ever required on that. And Alex mentioned one of the key aspects is its retail value is considerably lower than any sort of analogous fluorimeter, especially considering the analytics behind it and the opportunity to expand that into large networks of sensors, which can be valuable. Maybe next slide, please. Awesome. So getting a little bit more into the performance of the model itself, right now, the chart that I'm showing is an x-axis of the model estimated E. coli and lab enumerated E. coli on the y-axis, both in colony forming units per 100 mLs. So that red line would be a perfect fit. Exactly every model estimate is the same as the lab enumerated. And so based on our machine learning models right now, we get about less than a 20% error on both the testing and the training, meaning that the model is not overfit. Likewise, this is reflective of approaching other regulatory methods for enumeration of E. coli. It's notable to mention that the model is trained off of ground truth data, which is collected via sampling and those lab enumeration techniques, including membrane filtration and call alert, which both also have inherent errors between 20 and 30%. So this is, approaching it within that range, but also is trained off of that data. So it's important to note that we're definitely seeing anomalies between the data that we are enumerating between those different methods. So microbial enumeration and methods are still a moving target as far as direct quantification, which is something that this model recognizes. Next. And finally, I just wanted to review, as Alex mentioned, we have pilots that have been going on since March of 2025 in several different areas. We have a large focus in higher income countries on freshwater recreation, including a pilot with the city of Boulder in Chicago, as well as in Paris, France, and in Cleveland, Ohio. All of those contain water bodies that are highly recreated or aspire to be recreated. And so we're working in collaboration with community organizations and advocacy organizations, as well as city partners, to ensure support on regulatory compliance and real-time monitoring. Likewise, we do rural drinking water systems, predominantly in East Africa, which these sensors have been installed and piloted in rural areas near Kigali, Rwanda, as well as in northern Kenya. And something that we're more interested in further exploring is the interaction between the fluorescence mechanisms in different water contamination matrices, specifically brackish waters. So we have a current sensor deployed in Manchester Bay off the coast of Manchester. And then we'll hopefully be pursuing a pilot measuring the contamination that was aforementioned in a previous presentation on some direct sewage contamination in the Tijuana estuary.
Great, thank you, Whitney. This is our contact information, and obviously we're here for the Q&A as well. And as you heard, this is still pretty early on in our development process of this sensor. We've been working with about 20 units total this year, and we now have hundreds in production based on what we heard from partners and what we're seeing in the tests that Whitney just described. So an interesting time, certainly for our company, and we're on the lookout for folks like I'm sure many of you that may have a potential need or interest in distributed continuous E. coli sensing. So please don't hesitate to reach out if this looks like something that you'd like to try, because over the next year, we're just trying to find many applications, train a lot of data, and figure out if this is a niche that is really valuable for the water quality space. Thank you so much.
Awesome. Thank you both.
I'd also give one more plug if you guys are interested in the technology development itself. I'll put the paper that has preceded the design of what we know now as this sensor, in case anyone would like to refer to that.
Awesome. Thank you, Whitney. Certainly a group of us who like our scientific papers, so always good to share those. I'm gonna welcome Amy and John back on screen as well. We're gonna have about 15 minutes for a Q&A before we take a break and transition into our kind of more regional conversation and case studies. Folks are welcome to drop questions into the chat, which I know is already going on, but also feel free to raise your hands. I think maybe one of the questions from the chat we'll just start with as folks are thinking is, I saw a lot of kind of questions and discussions about as we eat animals, how that potentially changes markers in terms and our interpretation of results. I don't know, John or Amy, if either of you want to kind of elaborate on that.
Sure. Yeah. So no effect, right? Essentially, once you eat an animal, it's digested. What we're looking for when we look for source tracking markers for animals are bacteria that are common in their guts. And so it doesn't change the fact that the bacteria in your gut are still the same. They might shift a little if you decided to be a complete carnivore or something, but those general bacteria would still be there. So, if you were looking for ribosomal markers of an animal and there were undigested bits, maybe, but mostly, once you eat, the DNA from the animal is broken down into its constituents, and you basically use it, right? And the bacteria in your gut also use it as nutrients. So there should be no effect from that.
Great. I appreciate that, John. And maybe on a kind of tangential note there, do you want to just talk about the variability for humans in particular and what a non-detect might mean in terms of some of us have certain things in guts and others don't?
Yeah, so it does turn out that some people don't have the HF183 bacterium, the bacteria that harbors that genetic marker. In fact, I think in the Asian subcontinent, it's actually less common. But most people in North America do, and anytime you have municipal sewage, it's going to be there. So you might not, if you have an individual who is, clandestinely pooping in your stream from time to time that doesn't have it, you might not pick that up. But if it's from a group of humans, even, a few, it's likely one of them is going to have it. I think it's about 80% of people have it, if I remember correctly.
And then I was just going to add, I see some discussion in the chat that there can be false positives. So you can detect like HF183 or other human markers occasionally in other sources. And in the chat, dogs and raccoon were mentioned. That can happen, but generally when you have these kind of false positives, they occur at really low levels, much lower than what you're going to see in terms of concentration of like HF183 and other human markers when you have a human signal, like a true human source. And so I think just getting back to some of the fundamentals that both John and I talked about of understanding your land uses and when sources are likely. If you have a place you're not expecting human, but you see a bunch of dogs and you get a low level detection, then sure, maybe you should be concerned about cross-contamination, false positives there. So kind of contextualizing your results, being aware that there can be these really low level false positives, but also knowing that if you do have false positives, they're likely to occur at really, really low levels. And John, I don't know if you have anything else to to add
Yeah,
to
no,
kind of that point.
I think that's a really good point, and I would ask you to remember that dogs do eat diapers if you let them. They drink out of the toilet if you let them. So having some of those human fecal markers passing through them, even though they aren't the main host for them, that's not unusual. I don't think that's probably different than raccoons, who also are in your garbage. But there are cases of things like deer being a false positive source for HF183, possums. So it kind of goes back to what, is one of the tenets that we talk about in the source tracking manual and Amy covered, which is you need to know your watershed and what's in it. And if you have a lot of wildlife, you might want to test some of that scat if your marker data isn't making sense, and maybe try a different marker, right? And so like the HUM-M2 marker, for example, that Amy talked about, the EPA marker, that might be something you go to if HF183 was, you thought you were getting false positives, just to verify.
Right. Thank you both. Really appreciate the thoughtful reflections on that. And again, folks, feel free to raise your hand if you want to ask your questions live. Alex and Whitney, we've got a couple questions in the chat for you about just interference. So things like false positives from organic matter that's not fecal, potential for interference from the sediment side of things. Do you want to speak to that a little bit?
I can speak to the technical part first. Sorry, was trying to type out an answer in the chat, but it'll be easier to speak to like this. So absolutely, first of all, there are plenty of things that fluoresce that are not necessarily microbial or fecal matter. That's why we categorize it as what's called tryptophan-like fluorescence, so anything fluorescing in that range. However, the way that the model is calibrated tends to try to accommodate for that. And so using strong ground truthing methods are what we're currently doing to suss that out as much as possible. Likewise, literature has shown that there is so much of an extent to which tryptophan-like fluorescence and fecal indicator bacteria like E. coli are directly related to E. coli contamination. But all of that being said, one of the other important parts to recognize is that the sensor doesn't necessarily attempt to do direct enumeration, but rather categorize risk into what's actually meaningful. Counts of 50 CFUs per 100 mils versus 75 is not necessarily the valuable piece of information, but rather is your water within a proper range to be recreating or to be drinking. The second question was related to sediment affecting fluorescence, and this is absolutely also true. Sediment can quench fluorescent signals or increase light scattering and increase signals. Right now, that is accounted for both in the machine learning model as a parameter, but only from sources that already exist that measure turbidity. The newest iteration of the sensor that we're currently testing and validating includes what's called a time of flight sensor, which is essentially a mechanism that sends a beam of light and accounts for how quickly that light returns to identify whether the sensor is submerged in water or is left in air as a safety mechanism, and also to identify anytime a sensor might be running dry. And it acts as a quasi-turbidity sensor. So we're doing testing at the moment to identify how strongly we can identify different levels of turbidity and be able to quantify that in the model as well.
Right. Thank you, Whitney. And maybe just while we're on this kind of topic, there was a follow-up question about the graph you showed in terms of correlation to colony counts, and whether that 20% agreement is also at counts that low because of some of the challenges in low counts, whether it's field or lab duplicates.
Absolutely. So I can say one thing that's been incredibly challenging overall is identifying what that margin of error and level of detection is. On the ground truth methods that I mentioned that we use to establish that data, we see levels of error even within EPA and ISO accepted standards up to 20 and 30%, which at that low end can be even a concentrate or like an order of magnitude difference in concentration. So it is a little bit of a moving target, but on the tryptophan sensitivity side, we are able to achieve sensitivity of down to 1 ppb of tryptophan sensitivity. So the sensitivity level is there. It's more identifying the specificity, which is again why that target is focused on like the range of up to 10 CFUs per 100 mils, which is like categorized by the WHO as the low risk category for drinking water. So not necessarily a specific, we haven't necessarily distinguished a specific detection limit or anything, but it's definitely a challenge, but we do see a similar level of correlation at that low end.
And I just jump in, you know, part of what we're hopeful for with the price point and the business model, you know, is instead of finding partners to implement a single sensor, we would love to find places where 5, 10, 20, you know, sensors can be deployed so that this technology matches up really well with some of the source tracking that Amy and John presented on. So obviously the sensor alone, you know, one, isn't going to ever be perfect, and two, you know, doesn't solve any of the water quality issues itself, or do the source tracking. But if we drive the price down and we find where we can deploy at a watershed or a sub-watershed scale, and then we're also uploading data every 15 minutes, you know, not only can those sensors kind of help train each other on which data are outliers and should be kicked out, but also you have more granularity at a larger scale to really help on, you know, identifying sources and, you know, differentiating the different source types.
And I actually have to run, but thank you guys so much. Please message me with any other questions.
Thanks, Whitney. Appreciate it. So John and Amy, I think this question is more for you, which was just whether or not you see situations where there's kind of a mismatch in data between fecal coliform and microbial source tracking results. And if so, how you would think about that.
So where we see source tracking markers, but no fecal coliform?
That would be how I would interpret that.
Well, I have seen really low fecal coliform where there are source tracking markers, but Enterococcus was sky high, and that had to do with the source. And so it turns out seagulls have less fecal coliform than they do Enterococcus, but never. I mean, the reason we end up doing source tracking is because there's been a fecal indicator bacteria problem. And so otherwise, I mean, maybe you could look and then find it, but typically, you know, fecal coliform are pretty common in most sources. So I don't know, Amy, have you ever seen that?
Oh, Amy, you're muted somehow.
Hi.
There we go.
You're back.
Hear me now, cool. So I will just say, I wonder if this question is also addressing, like, it's hard to find correlations between FIB concentrations and MST marker concentrations sometimes. Like if you're looking for a statistical relationship between the two, that can be tricky. And so I think you are measuring different things, and these are biological samples. And generally, you know, you're taking two different filters. You have one filter for your MST markers, and you're incubating a different volume of water. And because you're measuring things that are biological, we know that they can be really variable based on kind of what volume of water you're grabbing, even if it's next to each other from the same bottle. And so I would say, like, when, you know, don't necessarily expect that like nice statistical relationship, but instead, like John's talking about, look for trends, like where you have sites that you have historical problems and you have lots of exceedances, you're probably going to have one or a few source tracking markers show up, even though those concentrations may not be directly correlated to each other, if that makes sense. And that's just because of all the variability that you can expect when you're measuring things that are biological.
Yeah, and you may have more than one source, so it's very possible to have high fecal coliform and see your whatever marker you're looking at for sporadically, because that particular source is not constant. So just like I was talking about that creek in Santa Barbara, they had leaky sewers and they had dog waste, and my guess is they probably had birds in the lagoon too, right?
The dog waste over the fence is a pretty memorable finding.
Yeah.
Yeah.
You read them.
Yeah. I was going to say, I know that there was also a lot of discussion in the chat about just the use of optical brighteners and the potential to combine that with caffeine as kind of a secondary supporting line of evidence. But just broadly speaking, anything either of you want to say about optical brighteners in this context?
We haven't had a lot of luck with optical brighteners. Mostly because of quenching from organic material. However, I do have a colleague who's retired now, Chuck Hagedorn at Virginia Tech, actually was able to use them very successfully along Long Island Sound, where there are a whole lot of septics. And so he put a flow-through fluorometer in a little boat. And during low tide, when the tide was going out, he just trolled along the shore, and he was able to identify several homes where the septic was flowing right through. And so he could see the detergent plume coming out of them. But that, of course, that's much clearer water than, say, the sediment-laden stormwater we usually get. We have not had much luck with optical brighteners for source tracking. Similarly with caffeine, the biggest problem we have is it's not really sewage specific. It's pretty much everywhere. So it's kind of like indicator bacteria, because any caffeine, which is in everything, you pick up. So it's not really specific to just sewage, right? Because, I mean, there are lots of people that might pee out in the open that drink coffee and Coke and energy drinks and such, right? So it's not very diagnostic. Other things, like some drugs that are used for epilepsy, they are actually pretty good. And so people have looked at those in groundwater, also ibuprofen. Right. So there are things that people have looked for with chemical markers, but we haven't done a whole lot of it. We have tried it to see if they match up with the source tracking markers, especially for when we're looking for sewage.
Appreciate that context in terms of things that have been tried and how it fits in with kind of other data as supporting lines of evidence. I'm going to do one more question just in interest of time, and then we'll give everybody a 5-minute break. I do see that there are some very specific questions in the chat about kind of detailed products, and we can maybe talk about those offline. But there was just kind of this theme of question around volume. And I know, John, you answered this in the chat, but just for visibility for folks, if you want to talk about kind of the theory behind the volume of samples and any tips there.
About how much volume? Well, the thing is you're limited by how much you can get through the filter. The way we do these assays is we catch the bacteria on a filter. And so depending on how much material is in there, even if it's a whole lot of bacteria, it will clog up the filter. These filters are, you can think of them as a sieve. They're polycarbonate, so they're a really thin piece of plastic. And Okay, so I'm going to get geeky here. The way they're made is they actually irradiate the plastic with gamma rays, and that makes weak spots in the plastic, and then they put it in a very basic solution. And wherever there is a nick in the plastic, the basic solution they put it in eats it away. And so the pore size is dependent on how long you treat it. And so if you look at an electron microscope, it looks just like the holes were drilled. Okay, those holes are about 0.4 microns, and so things like silt and larger bacteria and protists and toilet paper, all those things can clog them up. So you're usually limited with water that has a lot of material in it by how much you can get through it. However, that being said, in an ocean water sample, sometimes we can put 500 mils through it, right? And there you're dealing with the source usually being really dilute because the ocean things get diluted very fast. Typically, when a stream hits the ocean, it's not unusual for an immediate 10,000-fold dilution. So there, sometimes we actually take a dead end filter. It's used for kidney dialysis purposes usually, but we plug it instead of letting things flow through it. And basically, it's the right size to catch the bacteria, and then we back flush it, and sometimes we put 20 liters through it or more. So it really depends on your application, right? If you're doing sewage, you probably only need, a portion of a mil if you have like wastewater from the plant. But on the other end, if you have marine water, you need a much bigger sample. So you have to scale it to what you're trying to measure in your environment. And then the other thing with stormwater is sediment will clog it up. You put too much in it, you'll end up with essentially a cake of mud at the bottom. And then you need to use a soil kit to extract it. So this is all complicated.
I'll add just two quick things. One, I know that folks have questioned in the chat about reporting, and commonly you see these concentrations reported per 100 mil, and so that is normalized. That's not, like John was saying, how much volume you filter is very dependent on your site and how dilute you expect your sources to be, and how turbulent that site is as well. But it is common practice to normalize per 100 mil so that you can compare across studies. And then the second thing is just that John's talking about per filter, like I think both of us mentioned, being able to store and stash replicate filters is really important because you can kind of biobank these and go back and test other hypotheses without having to invest in another field study. So if you're thinking about your volume per filter, I just encourage you to multiply that by, as many as you can. You have time and capacity. Can you do 3 replicate filters? Can you do 5? You're going to be happy that you have those in the freezer. You're going to be able to go back and test a lot more things down the road, and these store pretty well for a long time. So.
Awesome. Thank you for that clarification and follow-up, Amy, on those pieces there. All right. I appreciate everybody's great questions. Geeky insights. All the geekiness is welcome here. We're going to take a 5-minute break.
Thank you, Marielle. Start my timer so I don't go over my 5 minutes. I'm Karen DuBose. I'm Skagit County's Pollution Identification and Correction Coordinator. I've been at the source tracking thing for about more than 10 years, actually. I've been at Skagit County for 10 years. I've probably been doing this kind of work for 15 or so. Next slide, please. So just some background about the area that we're working in. It's a very large watershed, 123 square miles. Also, something I forgot to put on the slide, it's also very rural. So we're not talking about houses really crowded. Maybe a house every 5 acres is the most crowded it gets. We've had known problems since about 2007. And in 2010, we had a downgrade of the Salish Bay shellfish growing area, which was a really big deal because we have 4,000 acres of shellfish growing area, and that's a lot to downgrade, and it caused everybody to kind of freak out. So we started doing a bunch of source ID tracking. Next slide. So we've done this twice. The first time was in 2010 when the downgrade happened. Everybody had lots of questions. Where's it coming from? It seems like I was not here at this time, so I'll add that. MST was a relatively new thing, I think, in this region. So the sites that you see in blue are our MST sampling sites. The red sites is just where our ambient sampling sites are. The sample sites all had very large catchments. So we're talking like mouths of creeks or like in the river, maybe several miles apart in the river. We sampled on an ambient basis. So, you know, just picking a day that seemed like a good day to go out. And we were looking for human markers, ruminant markers, gull, horse, dog, and bird markers. I don't have any information about how reliable those markers were at the time, so I don't know if there were opportunities for false positives and false negatives. I kind of suspect there might have been some opportunity for that. So let's go ahead and go to the results. The next slide. So what they found at the time was 58% of the sites had ruminant markers, 54% had avian markers, so birds are out there, not a big shock. 22% had human markers, only one site had dog markers. And then there were just other, they had a general bacteroides marker, and that was 57%. They did not find any markers for horses and gulls. Next slide. So this was helpful because we had a very vocal livestock agriculture community that did not believe that they could be the source, that it had to be septic, it had to be everybody else. And so to have more than 50% of the markers be the largest percentage be ruminant markers, that kind of shut them up. So we also were able to say we're working with everybody because we did have plenty of sites that had human markers. That said, there was little to no insight on where the problems were because each of the sites was, you know, like at the mouth of a creek. So great, somewhere in this creek, there's cows. In a rural area, there's cows everywhere. There's septics everywhere. So like exactly where is that? Next slide, please. So in 2019 and 2021, we had the opportunity to partner with EPA again. I will say that EPA did all the sample analysis for us, and we did all the labor for collection and shipping and whatnot. In this case, we focused on smaller areas. We've been doing source ID work for about 10 years, and we had several areas that we just have not been able to figure out after all these years. We know that our water quality problems are focused on storms, and so we focused on storm sampling. And that was awkward because storms only happen on nights and weekends around here sometimes, you know, and when you're trying to hit storms, it's really hard to find a sample when the lab is open and available and you have staff and all the things. And the markers we chose were cow, ruminant, human, and dog. Next slide, please. Results, we found evidence to push for enforcement at a few sites. So the map in the previous slide showed a really small area. I mean, some of these areas were like, there's just 3 houses. It's coming from one of these. We've looked at everybody's septic systems. We looked at all the things. We can't figure it out. We did get evidence to push for enforcement at a few sites, and that was really helpful. We found human and cattle markers scattered all over the watershed. What was really interesting is we found dog markers at every single site except for one. And so that really kind of flagged us that we needed to work on dogs. Another really interesting thing is Thomas Creek. It has really high bacteria levels in the summer. We expected maybe we would see septic markers coming through. We found no markers at all in the summer in Thomas Creek. So clearly there is something going on that we just don't have markers for. Last slide, please. So for the pros and cons, for as we see them for MST, it's really useful as a big picture tool. It's really helpful to kind of convince people that they might possibly be a source. It's a science that people seem to trust when you say you're doing DNA analysis, because nobody knows what MST means. They've seen CSI. They automatically believe that you're being honest. And then results from those small areas have really helped us narrow down potential sources. A con, the expense in labor and the cost of the analysis. The analysis, if we had paid for that second round, that would have been about $60,000 in lab costs, and we just don't have that in our budget. Storms are really hard to schedule when you have a lab staff that can't be available all the time, and results are often inconclusive, which is still frustrating. It was very frustrating to us to have done all of this work and only gotten a few small answers. And I am done, and only a minute and a half over.
Great. Thank you, Karen. More to chew on when we get to the panel. But I'm going to hand it over to Cameron.
Hey everyone, I'm Cameron from King County Stormwater. Next slide, please. Most of the source tracking work we do is in stream basins where there are bacteria impairments that are called out in our NPDES stormwater permit, but we do work throughout the unincorporated area of King County. We focus on looking for sources of human waste because of the human health implications. Really, this is more screening work than a study. Next slide, please. So we have found quite a few wastewater discharges to surface waters, over 25 properties since 2020, just through proactive screening, just going out in the world. The colored polygons on this map are stream basins called out in our stormwater permit: Bear Creek, Issaquah Creek, and the White River. Not very sophisticated. We use a simple approach of being mobile, covering lots of ground and measuring E. coli cheaply at many locations, currently using R cards. And then following up with tests for the human biomarker where we have repeated high E. coli results, and we do this in dry weather. The human marker results aren't always definitive, but they can provide important information when communicating with residents, our public health department, or the local sewer district. And you can see in the map that we have found a relatively large proportion of these wastewater discharges down near the White River on the Enumclaw Plateau. Next slide, please. So the streams on the Enumclaw Plateau have high levels of E. coli compared to other creeks in our jurisdiction. This is one of the most agricultural parts of King County. Lots of livestock. All properties are on septic systems except inside the city of Enumclaw. Poor drainage and high groundwater here, and that's bad for septic systems, but also for domestic animal waste. Next slide, please. Several dairies out here and many other properties with cows or other livestock. The owners of these animals often point to other possible causes of the bacterial pollution, not just septics, but also the local elk herd, which I will discuss in a bit. Next slide, please. So as I said earlier, our first priority is to find septic issues or to rule them out as much as possible. We have found quite a few, but even after fixing most of these, we haven't made much of a dent in the bacteria numbers in the creeks. And in some ways, that isn't super surprising if you consider the low flow rates associated with these septic discharges. It's still good to fix these, though. Next slide, please. So this bar chart just shows the frequency of the detection of the human, ruminant, and cow markers in the three main seasonal creeks of the plateau: Jones Creek, Second Creek, and Pussyfoot Creek, just from the last few years. Not a ton of samples, but what we see is that the human marker is detected only rarely, and where it is, we know that there are a couple of unresolved septic issues that we discovered through our screening work. On the other hand, the ruminant and cow markers are detected much more often. We've seen this throughout the years, not just the last few years. And what you're not seeing in this graph are the numeric results, the levels at which the marker was detected. Generally, our samples with our highest E. coli results in these streams also have our highest results for the cow and ruminant markers. That's not conclusive evidence that cows or ruminants are the main or only issue, but it is another bit of info. Next slide. So what about the other stream basins where we are focused? Bear Creek and Issaquah Creek that were shown on the earlier map. Those aren't agricultural basins, almost no cows, but lots of deer, a wild ruminant. And it's hard to know how many there are, but we see them all the time, just all the time. But the ruminant marker is almost never seen in these other stream basins. Next slide. So going back to the plateau, the elk, another wild ruminant, are known for coming down from the mountain in the fall and knocking over fences, just doing whatever they like. And they are often blamed by property owners for causing the bacteria impairments. I personally have only seen them once. I took this photo. We collected fresh elk feces from the Enumclaw Plateau, and with the assistance of our environmental lab, sent that material to a private lab in Florida, who developed what they called the elk Bacteroidetes marker. And then in 2 storm events, we collected 15 samples from 8 key sites in our stormwater system in the Boise Creek Basin. We analyzed those for E. coli, human, ruminant, and cow markers, and we also sent samples off to the lab in Florida for elk analysis. Next slide, please. So these storm samples had very high levels of E. coli, as you can see here. The ruminant marker was also found at very high levels. This is actually copies per mil. Next slide. The cow markers were also found at high levels. And a quick remark, the ruminant and cow markers seem to scale differently. Even in fresh cow feces, we get orders of magnitude more copies of the ruminant marker than the cow marker. And you can see down here that the elk marker was not detected in any of the samples. So does that mean that the elk are not responsible? No. There's a lot of questions we could be asked here, but this was just our limited attempt to entertain the claims made by some landowners, just hoping that having a little info regarding elk and also septics is useful when discussing these impairments with the landowners on the plateau. We looked into that. So the other markers that our King County Environmental Lab has available are dog and avian. We see the dog marker rarely in our water samples, less than 5% of samples for our stormwater programs, and that seems to be true for other county programs. Our environmental lab has tested fresh dog feces from a few dogs and found the marker present in all samples, so it should work locally. I could only speculate why we don't see it more. The avian marker became available just a few years ago. We haven't used it a ton in our stormwater programs, but the county's swimming beach program has analyzed a couple hundred samples from local swimming beaches for the avian marker. It's detected a lot more frequently than the human or dog markers. And it seems that the avian marker is often found at high levels in water samples, swimming beach samples that are high in E. coli. So that's interesting. It's hard to know exactly what it means. And sometimes it's hard to know exactly what to make of these results we get from these markers. They can tell us a little, but not always everything we would like to know. Next slide. Thanks to everyone, especially the King County Environmental Lab and the Public Health Department that helps us with this work. Next slide. That's it.
Great. Thanks, Cameron. All right, Cynthia, you're up.
All right. Hi, everyone. I'm Cynthia May. I'm the Water Quality Data Coordinator with the Whatcom Conservation District, and I'll be telling you about our Whatcom County MST study. Next slide. So you can actually go to the next animation. Oh, never mind. I guess the highlights aren't showing up. So our previous MST efforts that we've done here in Whatcom County are pretty extensive, going back to 1998. And I will just highlight really quickly that several of the earlier studies focused on marine sites only, and only in certain portions of the year, whether it was the spring season or 6 months. In 2016, our more recent study was a PCR study. It did cover the full year and focused on freshwater with 1 marine site. The drawback of that effort was that there were no local reference samples that were collected to confirm that our method could detect the sources in our local region. And then in 2019, I just wanted to share that Whatcom County, actually the Whatcom Conservation District, partnered with our local lab here in Bellingham, Exact Scientific, to do a fecal source reference catalog using whole genomic sequencing that looked at 29 species. And while this was, the first phase of method development seemed very promising, the tool definitely needed more development in order to serve as a confident source ID tool. Next slide. So our current study that we're actually working on right now, and I should say I haven't received data for yet, so I can't give you any nice looking graphs yet, is based here in Whatcom County up in the northwestern part of Washington. We're looking at 23 sites throughout the county, and these are our mystery sites, similar to what Karen was talking about, sites where we just haven't been able to find the source despite a tremendous amount of effort that has gone into finding the sources. The sites are cover 4 different watersheds, and the biomarkers that we're analyzing in cooperation with EPA Region 10 are 2 different human biomarkers, 2 cattle biomarkers, 1 non-cattle ruminant, and 1 dog biomarker. Next slide, please. So I'll just cover briefly some of the project improvements compared to prior efforts. First of all, we're using qPCR, which until the presentation today, I thought was the most helpful method to use, but now I understand maybe it's outdated and antiquated. So we think of it as a faster, more quantitative, and more widely validated, and most importantly, a tool that was offered by EPA Region 10, where they provided in-kind analysis services for this project. Next bullet. So we also are doing seasonal sampling. So we're collecting samples year-round, and we are also collecting extra samples during the seasons where we've seen higher counts from our fecal bacteria monitoring program. Next. And also the site focus has shifted slightly, so we're looking more at these smaller drainages with suspected sources rather than these really large river systems or marine systems that can be often quite diluted and can have a variety of bacteria inputs. Next bullet. And finally, this project, we're including reference samples to confirm that EPA methods work for our local bacteria strains, which was absent the last time we did this about 9 years ago. Next bullet. In terms of anticipated project benefits, like I said before, we're trying to identify all these mystery sources at these 23 sites that have just been chronically elevated and really challenging for us to track down the source for. Next bullet. So yeah, one of our benefits is to look at the DNA markers concentrations across time and space. So we have some of our sites are in series along a stream or drainage, so that we can look at kind of like the before and after inputs relative to certain non-point source or point source inputs. And of course, like I mentioned, we're looking year round and concentrating our efforts in seasons where there are high counts. Another benefit is, of course, we're hoping to be able to have some kind of interpretation for results when no markers are detected, whether that might be just kind of ruling out all these other sources and defaulting to wildlife as a probable cause, or just, yeah, having some way to understand when these biomarkers are absent from the results. And finally, and most importantly, probably we're evaluating the biomarker performance. So of course, we want to make sure that the EPA methods work for our local cows and septic systems and sewage treatment plants, and all the different potential sources that are coming here from coming from different sources here in Whatcom County. In terms of management actions, so one of our biggest management actions that we are working on here in Whatcom County is transboundary coordination, because we have several drainages that cross the Canadian border. We are, in close contact with our Canadian counterparts and meet with them regularly to help with source identification. And so any additional information we can gain from these transboundary sites and streams, and that we can communicate to our partners there, will just be really helpful for that additional source ID across the boundaries. And then, of course, there's, depending, and you can probably just bullet out all of these, depending on which hits we get, if any, for these different biomarkers, we'll have different kinds of management actions and follow-up. So if we have high human hits, we work with our local county health department to do septic system follow-up and maintenance. If there are high cattle hits, we will work with our state Department of Ag to do dairy follow-up if there are any dairies in the drainage. Dog, if there's high dog risks or hits, then we have a pretty robust pet outreach, pet waste outreach program that the county and conservation district work on. So we would potentially target more that drainage for more outreach related to pet waste management. If we get a non-cattle ruminant hit, we'll look for goat and sheep farms or owners in the drainage and follow up with them directly. If there are no markers detected, we're probably gonna we'll maybe default to wildlife and reach out to homeowners about keeping wildlife wild. And of course, the non-cattle ruminant goes beyond the goat and sheep category and includes deer and other wildlife, as Cameron talked about. And so that will be part of our conversation as we get the results in and figure out what our follow-up management strategies are. And I think that's it. We have a funding statement to thank EPA for their support. And yeah, I appreciate the time.
Great. Thank you, Cynthia. Well, and in trans-border kind of theme, we're going to hand things over to Natalie to share an update on GEMSTONE, which is an effort happening north of the border.
Fantastic. Thank you for the invitation to present today and to share a project that we've been working on here in British Columbia. I'm here representing the GEMSTONE project, and I am a microbiologist at the BC Centre for Disease Control. This is the equivalent of your state laboratories. And I'm also a faculty member at the University of British Columbia. And this project is co-led with Dr. Michael Lee and Lorraine McIntyre from our organization, Nico Prins, who is Executive Director of the BC Shellfish Growers Association, as well as Andrew Sheriffs, who is the Fisheries Coordinator for Malahat First Nation. I'd like to acknowledge that I am coming to you today from the unceded and traditional territories of the Coast Salish peoples, including the Squamish, Musqueam, and Tsleil-Waututh First Nations, as well as the Katzie Nations. Next slide. So I have for you two images of some challenges that our local fisheries encounter. So this project is very much focused on the shellfish industry and the challenges they have when their shellfish harvest areas are closed due to contamination. On the left, we see some large commercial shellfish fisheries where they are commercially generating or commercially processing shellfish. And when those become contaminated, those contaminated seabeds can be closed for many, many months due to a positive fecal coliform result. This has very significant negative impacts from a financial perspective, but also on public health when those oysters are associated with illness and just with the overall integrity of the industry. On the right, you see a more traditional harvesting of shellfish. This is Malahat First Nation. they are permitted to self-harvest in areas that are deemed closed by the federal government. However, how can they, in good conscience, make that suggestion if they don't have the tools to understand if the food is safe to consume, that traditional food? And so in both of these groups, both the large fisheries as well as the small ones, they have this challenge of this frequent closures without understanding the sources of pollution. Next slide. And this is really where this project comes in, to be able to use microbial source tracking and other data sources to understand the sources of pollution and to reduce the number of closures or the duration of closures. We have our main activities is to focus on assay validation. We've been involved in these studies before, but using commercial labs, this time doing the work ourselves. And we've coupled that with our traditional bacterial indicators as well as norovirus testing. We are doing longitudinal testing at 3 sites over a 1-year period, and we're looking at both biweekly sampling as well as surge sampling. We're coupling that data with climate data, vessel data, so boats, etc., as well as satellite imagery to understand the environmental variables, then integrating all those data sources together for source prediction and risk prediction. And ultimately, this project is about knowledge translation. It's not necessarily about the result, finding the source of pollution, but instead putting that knowledge into the hands of the producers. And in fact, for Malahat Nation, we are working with them to actually set up a lab so that they can actually have access to testing themselves, some data sovereignty when it comes to their own fisheries. Next slide. So some updates on the progress that we've made to date. Given the number of different interested parties, we really felt that it was so important to bring together a steering committee to direct the work, to inform the study plans and to interpret the data together. And we've met so far 6 times and have a 7th meeting coming up next month. We have done a comprehensive lit review on the ways that MST and fecal indicators have been used to mitigate and identify and mitigate sources of pollution. So we've relied on many of the great studies that the previous speakers have shared. We've completed the validation of, we selected our priority MST markers, as well as updated our norovirus assay for marine waters. We're nearly done our longitudinal and surge data, and we've been working on the geospatial data acquisition and integration. And at the bottom, you can just see some examples of heat maps with the various markers and what we're seeing in the different sites. So why are we doing this work? It was really about putting data into the hands of those who are most impacted by pollution. Here in Canada, the decision to close a fishery or to close a shellfish harvest area is done by a number of different federal agencies who are monitoring the environment, or because of outbreaks, in which case the BC government is involved in those closures. And it can be very, very frustrating to the producers not to understand, where the pollution is coming from, and in fact relying on a bacterial marker when in fact this is viral contamination. And so we're looking at a broader set of tools that can give them information of the contamination, norovirus specifically, as well as bringing in some of the MST markers as well as environmental data. The goal with this work is to have stronger food security in general, but more specifically, stronger Indigenous food security. And ultimately, we're working towards protecting public health by reducing the risks of foodborne illnesses specifically related to norovirus and shellfish closures. But we know that this can then be applied to other applications. For example, we have a number of beaches that are closed every summer without the knowledge of where that contamination comes from. Why do we have these recurrent beach closures? So we can see the other applications downstream. And lastly, we know that this is going to result in economic savings. Next slide. With that, I'd like to acknowledge the extensive research group that we work with, but also the number of different funders that have been involved. And I think this shows the interest and the power of this tool and the potential of this tool. And with that, I'm happy to answer any questions, but sharing with you my email in case there is some interest in cross-boundary collaboration.
Thank you, Natalie. Always cool to see examples of that sort of data sovereignty side of things. So love that that's been centered up front. I'm going to invite all of our presenters back on screen for our panel discussion. Thank you all for the lightning talks there, just to give a sense of context and flavor for the conversation here. I welcome folks to raise your hand, and that'll allow you to come off mute and ask a question of our panelists. While folks are working up that courage, I guess one question that's been touched on briefly across these is just the difference in terms of urban versus rural settings and how you think about source tracking in that context, but particularly in the application of microbial source tracking. So, anyone want to take that?
I can talk about it. In urban settings, typically we don't deal with, we don't have to deal with cattle. Dogs and humans are the main issue. So it makes it somewhat simpler, but the built environment is also somewhat more complex in that you have a network of storm drains and sanitary sewers running underneath. Especially in Southern California, we don't have combined sewers. So and so it's interesting, the stormwater agencies, when there's fecal material in their storm drains, say, hey, we don't do fecal material, it's not us. And the sewer agencies say, our lines don't leak. By law, they can't, and so they don't. And so you run into that, and you actually have to show them proof, which we've done now by actually doing empirical measurements of the leakage from some of their lines. Typically, and probably in Washington, where you all are, because of the rain, infiltration into sewer lines is probably something that folks worry about more than exfiltration from them, because the sewer agencies don't want to treat water that isn't sewage. So they're very fastidious about that. But yeah, there are some big differences, and I was thinking about talking about the cattle in the streams. There was a study I was involved in Virginia, where we continued to see high fecal contamination, fecal coliform, in this one stream, and there were cattle around, and they swore their cattle weren't in there, and we found cattle markers. And so we did an experiment and put up a fence, and all of a sudden that section of the stream, we didn't have the problem anymore. So the cattle definitely do get in the streams, and they poop wherever they are. So yeah, a lot more different challenges and different spatial challenges in rural and urban settings. I know Amy's actually worked both places too.
I'll just throw in there that usually rural areas properties are on septic systems. So everything else being equal, the material, the fecal material, is going to take longer to get to your sampling point from the organism if it's passing through the ground first, as opposed to like a sewer cross connection. So that travel time, and also the weathering or damage that the genetic material might experience as it's traveling through the ground, this material, the genetic material, doesn't hold up the way that the E. coli bacteria do. So that could be an issue when you're looking at your results for your human marker.
Great points on those. Thank you both. I see Andy has a question, so we'll go to you, and then Kia after.
Yeah, thanks. And thanks for all the presentations. I think this was I haven't been following the chat, so you may have addressed this, but Cameron, when you were showing your cow versus elk versus ruminant markers, like some of us have been talking about the specificity of those marker sets, and wondering if your testing included like how much I guess confidence you have that an elk is an elk and a cow is, and ruminant kind of covers them all. Like did you guys do enough testing to to give yourself a pretty decent set of is your confidence pretty good on those?
Well, I'll just tell you what we've done. I mean, the
Sure.
elk feces were collected from the Enumclaw Plateau. The North Rainier elk herd, the part of that herd that comes down onto that plateau is pretty small, probably 100 or less individuals. Now, I don't know, the private lab in Florida that we sent that material to, that's a bit of a black box, and we had some other issues with that lab. So thanks for asking. Just taking the results at face value, they developed the marker and they didn't detect it in our water samples. But yeah, I don't know. Yeah, beyond that, I don't know what to say about that elk marker. But it was from the local environment. We have harvested cow feces from King County, and just done that a small number of samples. Okay, like a few to several, and the cow marker turned up in all of those, as did the ruminant marker. So again, it's kind of at the level of an anecdote, but we did at least test local fresh feces to see if it got hits for our those markers. And so maybe that's all I can say. Does that kind of answer your question or address it? You're muted. Oh,
Marielle didn't give me permission to unmute, so you get one question and then you have to be quiet for the rest. So anyway, yeah, no, that's helpful. I appreciate that. Thanks.
Well, and I think building on that, one of the things that stood out to me is just this idea of the duplicate markers that many of you have touched on, and the value of that. So, all right, here, let me hit the magic button and you should be good to go now. Oh, no, still not hearing you. So there you go.
Yeah. So 2 questions. One question, I heard crossphage was mentioned as another marker for testing human contamination. And I wanted to ask, how confident are you of crossphage? How difficult it is to measure? I know you need more volume because it's a phage. So if you could, that was one question. And I'll ask the second question after I hear the answer on this.
Okay, well, it seems to be pretty specific. We have not tested it across a lot of different samples yet, but we've tested it in the urban environment, and it seems to correlate pretty well with HF183. The way we actually test for it is you can't easily use a 0.02 micron filter and get much water through it. So the way typically we do is we used a charged membrane, and so a nitrocellulose membrane with magnesium chloride acidified to pH of 2 seems to catch them relatively well. That's true for most viruses. And so we are doing a study right now where we're measuring HF183. So I should back up. We did a study before where we measured HF183 at a bunch of watersheds to see how much of a component it was. But the question we have is, how much does it get diluted when it gets to the beach? So we're redoing that study, doing above the tidal area in the stream or storm drain and at the beach in the mixing zone. And we've included crossphage this time. And so we'll actually have data on that probably spring next year. And so you can look for a publication and then we'll see how it compares. But we've had good success finding it.
I think it's good to have 2 markers so that you could get better correlation.
Well, I would agree. And the other advantage is it is itself a virus. And so bacteria and viruses don't behave the same in the environment.
Right.
And so it's important to us to know
Yes.
if there's a virus-like particle like crossphage that gets somewhere, that means that the human pathogens are also getting there. Yeah.
And how much volume typically you would test for crossfudge? 250 ml?
If we can get it through the filter, yes, 250 mil is fine. 100 mil if we can't. And the other thing is you've kind of got a trade-off depending on how many other things are in the water, because you can actually filter too much water and get inhibitors in it, and then you lower your yield. So you're always playing that game.
I was just going to add, if it's okay, I dropped a study that was done by a group in Canada where they actually used crossphage to look for septic effluent, and it was more successful. They didn't detect HF183, but they could detect crossphage because it was groundwater derived, and we know that viruses move really differently in groundwater than bacteria. So I'm glad you brought up crossphage, and as John said, it's just like a really great tool, and because it's DNA based, it's not as complicated as some of the RNA based viral assays. So I dropped that study. It wasn't done by anyone on this call. It was just one that I thought was like a really cool application that might be helpful when thinking about crossphage and kind of how it compares to HF183, and when there are applications where it's a particularly promising tool.
Another quick question. I've been asking about how to report the volume, 100 ml, so that I know you can compare across labs, you have a benchmark, but we typically filter 400 ml, and what is found in 400, especially at the lower limits of detection, may not be found in 100 ml. So I am, my past histories from US EPA, so some of my EPA colleagues back in Cincinnati told me, just report how much you are filtering, which is 400 ml and not 100 ml, because it may not be there in 100 ml, but 400 ml. And, you know, just your thoughts on it. I know 100 ml is the standard, yeah.
Okay. Well, my thoughts on it are, yeah, that's a good point. I would probably still report per 100 ml, and then I would put something in the discussion to talk about the fact that I'm filtering 400 ml and make your point that it may be different, but that you're reporting 100 ml, because that's how most water quality folks think.
Right.
Great. I also see a question in the chat just about, now I lost it, sorry. What GCN you would consider elevated to trigger an investigation? If anyone has thoughts on that.
My brain isn't translating GCN. What is GCN?
Gene copy number.
Okay.
And I thought 485 was mentioned in one of the first slides.
So John can speak probably to this too, because he actually led one of the studies that I just mentioned. I said, you know, that where the research community on is that there's kind of a nice framework now for developing risk-based thresholds for these markers, using quantitative microbial risk assessment as the tool. And there's been a couple really good studies that have come out, and so far these kind of benchmarks have primarily been quantified for HF183, since that's widely used human marker, and they've ranged between about 100 in the literature to 500. So it depends like what you're sampling, you know, if you're sampling effluent versus if you're sampling at a beach, because we expect that HF183 signal to act differently than the pathogens that actually make people sick. And so that's why there's kind of this difference. But in terms of order of magnitude, at least the papers I've seen, it's like 100 to 500. And then John and Squirp led a really interesting study where they did that in stormwater. So I don't know if he wants to talk to the particulars. And then the slides will be shared
And I linked out the 3 papers that have taken that approach, so you can look at those.
Yeah, so the body of evidence, and kind of winnowing down to a number is growing, but because it's not a regulated thing, typically it's whatever the regulators in your area will tolerate, right? So we have some folks in San Diego that it's like, if there's any human marker at all, they want an investigation and really push for it. And so it really varies by region, but typically 100 is a pretty good rule of thumb. Consistently, right? If it's once in a while you see 100, maybe that's iffy, but it's not a very big source of contamination, or at least it's a pretty degraded one if it's below 100.
I have a little different answer. I mean, when you say 100, you're talking about gene copies per 100 milliliters.
Yeah.
Our lab has set our MDL for our human marker, which is based on HF183 at I guess the equivalent of 110 gene copies per 100 mils. I think they technically could report lower numbers, but and if any of them are here, they could speak more eloquently than I can about it. But there could be some noise at the higher, at the end of the cycling process that is generated, during qPCR. We generally, if we get a positive result of less than 1,000 gene copies per 100 mils, we don't really react very strongly, because we have limited resources, right? We're trying to give ourselves the best chance of actually finding a problem, and not going on wild goose chases. So generally over 10,000, we think of it really as a confidence, it's more of a confidence thing rather than a quantity thing. If we get a result of over 10,000 copies per 100 mils, we're like, we have strong confidence there's something going on. We need to do more here. Below 1,000, meh, and then it's sort of in the middle. It kind of depends, what other information do you have? And yeah, definitely you want to get more than one sample before you pursue anything, I would say. More than one hit. So
I'll just add, Cameron, I think you bring up a really good point that there's like 2 ways to think about how you're interpreting these results. One is like, where could this be a potential human health risk? And those are the thresholds and threshold development I think John and I are talking about. And 2, you're thinking about like utility of using it to track down fixed sources, and how do you actually prioritize where to start. And so that application makes a ton of sense, like starting with where you have really strong confidence in the number that you're going to be able to actually track down that source and fix it, starting higher than kind of like these low levels where we think there might be some increased risk. Theoretically, it makes sense. So just wanted to call that out, that I think there's like 2 ways to interpret data, and depending on how you're using it, it's that's gonna be important for really what level is kind of triggering action.
Yeah, the levels Amy and I were talking about are Rec 1 exposure. All right, so you'd be swimming in the water.
Appreciate the thoughtful discussion on this. Another question that I've seen is, and the theme that comes up, is just the cost of this sampling in particular being a barrier to using it. And curious, particularly for folks in the Puget Sound region, if you see opportunities to work with lab partners to make these microbial source tracking analyses more affordable. Or I should say Salish Sea, sorry, Natalie. I don't know, Karen, Cynthia, if you've seen things in other spaces in terms of lab collaborations in our region that you think should be considered here, or if that's more just a reminder that with cost about where this fits in terms of when you do microbial source tracking.
Well, I mean, we collaborated with EPA, so we didn't pay for any of the analysis. And I think in the original 2010, we collaborated with Kate Field at OSU for a small number of samples and then EPA for a larger number of samples. So I don't think we paid for any of that analysis, and it's a huge chunk of money. I mean, if you have somebody willing to pay $60,000 in sample analysis, by all means, take advantage of it. But I think, and I think Cameron might agree, we've chatted about this, you can take an awful lot of samples, like regularly, especially with the cheaper tests doing source ID, and get closer. Yeah, EPA Manchester Lab is who we've worked with, and they're wonderful to work with. It's just that the answers aren't. I think we all want the magic light switch, right? You know, the thing that tells us the answer, and none of us are getting that. It's frustrating being the person on the ground and having people who want. They assume because they see, you know, they watch TV, they see CSI, they know DNA works. Why can't you tell me the answer? You know.
Yeah, and I'll just second the EPA Manchester Lab. You know, we're in the process of working with them right now. They've been an excellent resource helping us guide our project planning and, you know, co-op development. And I think at one point I calculated how much they were saving us by running the qPCR samples in kind, and it was like hundreds of thousands of dollars. And so it's significant. I mean, we're doing a year-long study. We're sampling 23 sites, some of them multiple times a month. So it's made that work possible. And in my conversations with them, they've been open to doing, you know, they're always open to entertaining other ideas, whether small or large projects. I very highly encourage people to reach out. And I can put the contact person in the chat, and maybe she won't want me to do that, but I will anyways.
It's cool to see those collaborations that really make things possible in terms of a lot of this work in these studies. So that's great. And Cynthia, I got a question about just for what you all are doing. Can you remind me whether or not that developed reference samples to be evaluated for unknown samples?
Say that again. Are you talking about our current project or a past project?
Yes. And the question was whether it's developing reference samples to be evaluated in unknown samples was the clarifying question.
The reference samples were taking actual known fecal bacteria sources. So we collected from wastewater treatment plants, septic systems, and directly from dogs and deer. And yeah, to all of trying to validate the biomarker methods used by EPA. So those were to confirm that known sources were picked up in the methodology.
Thank you for confirming. Okay, so another theme that I hear come up in a lot of conversations as we're thinking about kind of the management options is the difference between Canadian geese that are coming through in a migratory context versus chickens, and kind of the bird side of all of this. Curious if folks have any kind of reflections or thoughts they want to share on that front, broadly speaking.
I'll say that when we were working with EPA to develop our second project, we wanted to use the bird marker, and they recommended that we not, because birds are legit everywhere. And they said, you know, you're going to find bird markers everywhere. So without a specific marker for winter birds is a big issue here in Skagit. So without a specific marker for trumpeter swans or, you know, one of those birds, it's not going to be useful for us. So we skipped it.
Thanks, Karen. Appreciate that. Any questions you all want to ask each other? Just because you're on the panel side doesn't mean you can't ask questions as well. Thank you, Cynthia, for dropping that contact into the chat. Okay. And it may be that we're just hitting a point of saturation for everybody's brains on a Friday morning, in which case that is a-okay. So I'm gonna give folks one final call for an opportunity on questions, and just remind you that we will be sharing both the recording and the slides out to everybody who registered, and we'll also post those on our website for ease of access. But hope that today kind of prompted some new insights, some different ways of thinking about how microbial source tracking might fit into the work that you're doing. And maybe some new connections in terms of folks who are doing relevant work either in our region or more broadly, that there may be opportunities to collaborate with. But final call for our panelists. Any final thoughts that you want to throw into the mix related to microbial source tracking or just management of fecal coliform more broadly?
Marielle, from a practitioner standpoint, I'm wondering what the best way is to stay abreast of all the new technological developments. And there's a lot of things, there are a lot of things happening in this world. And, sometimes it feels like things are progressing really slowly, and then you check out for a little bit and check back in and everything's changed. So I don't know, Amy or John, do you guys have suggestions for email lists or organizations that would be good to kind of follow or just kind of keep in touch with, so that we can see when there's new opportunities to maybe incorporate into our work here at the municipal level?
Well, I mean, you could certainly check our website here at SQUIRP, but I think probably where we're headed next is another method evaluation study. So that last method evaluation study was over 10 years ago. I don't really want to do another method evaluation study. There isn't really money to do one right now. They are on the order of about $1 million to do what we did last time. But if there are a whole bunch of markers out there that people are using and we need to have confidence in them, then that's something that we should think about doing. So I think a continual check-in as the technology continues to evolve. I do think CRISPR is going to be the next single marker kind of technology that gets used, but I think sequencing is also going to be a big thing, especially as it gets more accessible, and we can do long read sequencing of whole communities. That's going to be important, and even ribosomal markers that target the animals that are shedding themselves are coming into vogue. So things are moving. I just think right now the research climate is terrible. Folks' funding has suffered a lot, and so it's going to take a little while to recover before we start seeing things push forward. But I'm still going to try to steal the technology from my folks in the biomedical field and co-opt it to my evil purposes.
I was trying to think, that's a good point, that there's kind of a gap in terms of listservs and other information to make this, just to get those updates easily. But the National Water Quality Monitoring Council, I think they have annual conferences, and they have a lot of, EPA has a great web page, and you can even just look at who's talking and what they're talking about if you can't attend, and reach out to individual presenters. I think that's a good conference to keep an eye on. And they have a conference, but they also have a group, and you can kind of sign up for different work groups, and that's water quality monitoring more broadly. But there's a lot of people who participate who are interested and involved in MST methods.
I was trying to think the UNC conference, but that hasn't happened in a while. Like John mentioned, there's just a lot of these things are on pause that we could normally recommend, but I do think the National Water Quality Monitor Council is a good one and kind of can act as a good hub.
Yeah. One thing that's going to keep happening and what pushed things in that study I talked about in San Diego is folks are coming to the end of the TMDL process in their watersheds. And so that study was actually a study to look at all the human sources in that watershed, which we did. I just talked about the biofilm part. I think that's going to happen more and more where folks are getting an investigative order, and then that's going to push more things to get done, and that tends to free up local money to do this kind of work. So we're not dead in the water. We all still have our goals that we have to meet. And so I anticipate that it will move forward, even though there's not money for just research. There's going to be research done because there's a need for it to solve some of these problems, and that's going to continue to push technology forward.
Great, appreciate those suggestions in terms of places, and as PSI, we can talk with Leah and others at the SIL and see if there are perhaps some regional forums on an annual basis whereby we can share some updates as well. And I think just in that theme of kind of forward looking, we'll highlight again Amy's paper on the nutrients and fecal source tracking in conjunction, just because I know that some of the voluntary guidance that ecology's put out recently is put an increased emphasis on PIC programs looking at nutrients, is my understanding as well. So just thinking about trajectories, think about that component as well on as the technology piece.
Yeah. No, and as a side, just being a microbial ecologist, nutrients themselves can cause bacterial blooms with just a little bit of seeding. If you're not finding source markers and you have high fecal coliform and you can't figure out where it's coming from, you might want to look at the nutrients. It only takes a little bit of E. coli to start growing and be very happy if they have what they need.
Good to know. Appreciate that, John. Well, thank you all so much. Again, I know folks are coming back from maternity leave. Folks are leaving for vacation this afternoon from our presenters. And so just appreciate all the time and thoughtfulness that went into sharing these insights and the great questions in the chat from everybody. So thank you. And I hope everyone has a lovely weekend.